Video lecture, we will see about how the urine formation takes place. Now, the urine formation takes place, or the structure that is involved in the urine formation is the nephron. Okay, inside one single kidney there are around 10 lakh nephrons. Okay, so in both the kidneys, okay, the glomerular filtration rate that we studied in the previous video lecture is that in both the kidneys per minute 125 ml of the filtrate is formed, which is called as a glomerular filtration rate. Throughout the day, in one day, 180 liters of the filtrate is formed, but this 180 liter is not excreted in the form of urine. So what happens is that we are going to see over here. Okay, now the process of the urine formation shows ultrafiltration, reabsorption and the tubular secretion. At the end, how much urine is going to form is about 1 liters to 1.5 liters of the urine is excreted throughout the body per day. Okay, so 180 liters of the filtrate is formed, but from 180 liters only 1 to 1.5 liters of the urine is excreted from the body. That happens because of the process that is called as a reabsorption. Okay, now there is ultrafiltration, there is reabsorption, and then there is a tubular secretion. Well, let's see what is basically the ultrafiltration. Ultrafiltration is a process. Okay, we know that there is a Bowman's capsule. Inside the Bowman's capsule there is a tuft of capillaries which is called as a glomerulus. Okay, from the afferent arteriole the blood is going to come with a pressure. That pressure we studied, that is around 100 mmHg. Okay, so the blood comes with the pressure and it falls into the glomerulus. From the glomerulus the filtration takes place. The pressure, okay, by which the blood is coming, and then there is also another pressure that is called as the glomerular blood hydrostatic pressure. Okay, we saw what is the net filtration rate. Because of this, the filtration takes place, and that filtration is called as the ultrafiltration. Because of the filtration, many of the substances are filtered and they come into the Bowman's capsule. Now, what are the substances that come into the Bowman's capsule? Water is a larger portion that comes into the Bowman's capsule. Along with water, whatever is present in the blood except proteins and fats. Okay, proteins and fats cannot be filtered. That you have to remember. Proteins and fats cannot be filtered, so they are not a part of the filtrate. They do not come into the Bowman's capsule. But other than proteins and fats, let it be glucose, amino acids, hormones, vitamins, ions, okay, sodium, potassium, chloride ions, okay, drugs it might be, or it might be different types of hormones. All this comes into the Bowman's capsule and becomes a part of the filtrate. Okay, so this process is called as the ultrafiltration. Now this ultrafiltration, if we study, okay, it is under three control. One is called as the myogenic control. What we are talking about? We are talking about the regulation of ultrafiltration. Who is regulating the ultrafiltration? Myogenic regulation. Myogenic regulation means this myo means related with the muscles. This afferent arteriole. Okay, what happens is that the muscles of the afferent arteriole they get contract, and when they get contract, the diameter of the afferent arteriole is going to reduce. When the diameter reduces, the pressure or the flow is going to increase, and because of that the blood comes with a higher pressure to the glomerulus. That is what is going to happen. Okay, so that is the myogenic control. Second is called as the juxtaglomerular control. Now what is the juxtaglomerular control? We studied about the juxtaglomerular apparatus. Okay, we saw that there are some cells which are called as the juxtaglomerular cells, and there are some cells which are called as the macula densa cells. All these together they are going to form the juxtaglomerular apparatus. This juxtaglomerular cell secretes an enzyme which is called as renin. Renin is also very much responsible for maintaining the pressure and the blood flow, whether it might increase the blood flow or reduce the blood flow. Okay, absorption and reabsorption, and that is how the glomerular apparatus is playing an important role in regulating the ultrafiltration. And the last one is the neural control. So there are three controls. Third one is the neural control. Now the kidneys, okay, and both the kidneys are innervated. That is, the neurons of the sympathetic nervous system enter inside the kidneys. Now when the sympathetic nervous system is activated, that is in an alert condition, okay, when we are scared or when we are under some pressure, okay, so at that time the body becomes heightened, okay, all the activities inside the body becomes heightened. But what happens with the excretory system is that the sympathetic nerves, what they do is that they carry out the constriction of the arterioles, and constriction, when it happens, the blood flow is going to reduce. Okay, so that is what is going to happen with the sympathetic nervous control. So in case of the myogenic, the blood flow increases. Okay, in case of the juxtaglomerular apparatus, it is going to maintain the blood flow and the glomerular filtration rate. And in case of the neural control, the sympathetic neurons which have innervated the kidneys, they are going to reduce the blood flow, and thus they are going to reduce the glomerular filtration rate also. So these are three controls. One is myogenic control, the second one is the juxtaglomerular control, and the third one is called as a neural control. Now after that, when the filtrate comes into the proximal convoluted tubule, what happens? Okay, we are going to the point of reabsorption. What happens over here is that 65% of the filtrate is reabsorbed back. Now this is a nephron. There are 10 lakhs of nephrons in a kidney. In between the nephron there is some space, the medullary space. That medullary space is called as the interstitial space. And these nephrons are surrounded by the blood capillaries, which are also called as a vasa recta, and peritubular capillaries, which are in contact with the proximal convoluted tubule and the distal convoluted tubule. So reabsorption means whatever the filtrate is formed in the proximal convoluted tubule, most of the substances are taken back from the PCT and they are poured into the vasa recta. Okay, that is called as the reabsorption process. Now what happens is the proximal convoluted tubule, 65% of the substances, water is reabsorbed. When the water is getting reabsorbed, the process is osmosis. From that is, the water is going to move from its higher concentration towards its lower concentration. What happens with sodium, potassium and chloride? Sodium are basically the ions which are reabsorbed by the active transport. This you have to remember because questions are many times asked on which type of transport does takes place. So sodium is basically by active transport. Then when we talk about the chloride ions basically they come out by the process of diffusion. Urea and some other solutes, they also are reabsorbed by the process of diffusion. Water is by osmosis. Okay. Then when we talk about glucose and amino acids, almost all the amount of glucose and amino acids are reabsorbed back. Okay. So glucose and amino acids are reabsorbed by the process that is called as the secondary active transport. Okay, and then what are remaining are the amino acids and glucose are reabsorbed, sodium, potassium and chloride are also reabsorbed, hormones are reabsorbed, vitamin C, okay, water soluble vitamins like vitamin B, they are also reabsorbed. Now after the reabsorption process happens, what happens over here is that whatever the filtrate is formed, the filtrate okay, is going to get transported to the loop of Henle. So when it comes into the loop of Henle, what is going to happen? Loop of Henle is basically loop of Henle, that is the descending loop of Henle is basically permeable for the water molecules, but ions are not going to get transported from the descending loop of Henle. So water is reabsorbed into the vasa recta, but the sodium, potassium, chloride ions, they are not reabsorbed, and because of that here inside the tubules, the osmolarity of the filtrate becomes hypertonic, okay, as compared to the osmolarity that is present in the blood plasma. So here inside the hypertonic solution is maintained. Now from here the filtrate enters and goes towards the ascending loop of Henle. This ascending loop of Henle is not permeable for water. So water will not be reabsorbed. But all the ions and solutes like sodium, potassium, calcium, magnesium, all will be reabsorbed into the blood plasma. And because of that, here the concentration of water is going to increase because the solutes are absorbed. So here, as compared to the blood plasma, the filtrate, it is going to be hypotonic in nature. The osmolarity is hypotonicity, or it is hypotonic in nature. Next, all the filtrate, then it comes towards the next tubule, that is the distal convoluted tubule. In distal convoluted tubule, you have to remember that two hormones are going to play a very important role. One hormone is aldosterone. When the sodium ions has to be transported, aldosterone plays a very important role, and under the influence of aldosterone, the sodium is reabsorbed. Then the chloride ions are also reabsorbed, and the another hormone, that is antidiuretic hormone, that is ADH, which is basically secreted by the posterior lobe of the pituitary gland. Under the influence of that is the antidiuretic hormone, the water is also reabsorbed. Okay. Then finally here again it becomes isotonic in nature. Okay, that is the filtrate that is present inside the distal convoluted tubule, it becomes isotonic. From here it goes to the, that is the filtrate goes to the collecting duct. Collecting duct also, under the influence of aldosterone, absorbs sodium ions, and it is permeable to water. So that's why collecting duct, from the collecting duct, the water is also reabsorbed. And after reabsorption of water, whatever the filtrate is formed in the collecting duct, it becomes hypertonic as compared to the blood plasma. And here when the filtrate comes, it is now called as urine, and urine is hypertonic as compared to the blood plasma. So this is about the reabsorption process that is taking place in all the different sections of the nephron. Okay. The filtrate comes over here, around 1100 to 1200 ml of the filtrate is filtered per minute, and how much or what is the glomerular filtration rate is 125 ml of the filtrate is formed per minute. Throughout the day, 180 liters of the filtrate is formed. From that 180 liters, only 1 to 1.5 liters of the urine is excreted from the body. The remaining part is again reabsorbed. Reabsorption means from the tubules, all the substances are going to be poured, okay, into the vasa recta or the blood capillaries, so that they can remain inside our body and they should not be excreted outside the body. So in the proximal convoluted tubule, 60% of the substances are reabsorbed. Then it comes to the descending loop of Henle. What is reabsorbed in the descending loop of Henle is major amount of water. Ions are not reabsorbed. Then the filtrate goes to the ascending loop of Henle. Ions are absorbed but water is not reabsorbed because it is impermeable to the water molecules. Then it goes to the distal convoluted tubule. In the distal convoluted tubule, sodium, chloride and the water molecules are reabsorbed. Sodium is reabsorbed under the influence of aldosterone hormone, and water is reabsorbed under the influence of antidiuretic hormone. Finally, the filtrate goes into the collecting duct. Again, in the collecting duct, sodium is reabsorbed and water is also reabsorbed, where the urine is formed, and urine is hypertonic as compared to the blood plasma. Now, the next point that we will study is the tubular secretion, which is the last part of this point, that is the ultrafiltration, reabsorption and the tubular secretion. You have to remember that tubular secretion is exactly opposite of that of the tubular reabsorption. In reabsorption, from the tubules, the substances were poured into the vasa recta, that is the blood capillaries. Okay, but here what is happening is that from the blood, now all the things, or from the interstitial fluid, that is a fluid that is present in the medullary region, all the substances, unwanted substances which has to be excreted, okay, that are poured into the tubules so that they go to the collecting duct. In the urine they will get mixed, and from the urine they will go to the urinary bladder. Now, when we talk about the proximal convoluted tubule, in the proximal convoluted tubule, what is secreted, that is what is taken into the proximal convoluted tubule? Hippuric acid. We know that hippuric acid is another nitrogenous waste which is formed by the combination of benzoic acid and the glycine. So hippuric acid is secreted into the proximal convoluted tubule. Creatinine. Okay, we know that creatinine is basically produced in the muscle cells. That is also secreted in the proximal convoluted tubule. Ammonia, nitrogenous waste which is formed because of the deamination of the amino acids, that is also secreted into the proximal convoluted tubule. Finally, pigments. there are different pigments like bilirubin, biliverdin, then urea also has another pigment which gives yellow color to it. That pigment are also secreted into the proximal convoluted tubule. Now from the proximal convoluted tubule, these many things are secreted in the PCT. Next is in the ascending loop of Henle. The thin part of the ascending loop of Henle, in that urea is secreted. That is, the urea from the interstitial space, it is collected into, or it is secreted into the ascending loop of Henle. Next is in the distal convoluted tubule, what is secreted? Bicarbonate ions, okay, then ammonia ions, then potassium and hydrogen ions, they are also secreted in the proximal convoluted tubule. Now as the ions, okay, like potassium, hydrogen ions, bicarbonate ions, all they are secreted in PCT and DCT. And because of this secretion, what is happening is that the blood pH is maintained between 6 to 8. Okay, so this reabsorption or secretion is basically also playing a very important role in the osmoregulation or the balance of the ions and the balance of the pH of the blood. So finally, in the collecting duct it goes. Now what happens in those animals whose glomerulus are not properly developed? Okay, for example, amphibians, reptiles whose glomerulus are not, or fishes you can take, whose glomerulus are not properly developed. In those, tubular secretion plays a very important role for the formation of the excretory product. So you have to remember that. In humans, glomerulus is basically properly developed, Bowman's capsule and nephrons are properly developed. But what about those organisms where the nephrons are not properly developed? In those organisms, for the excretion and to maintain the osmotic regulation of their body, or the osmoregulation of their body, those organisms, excretory organs, are completely dependent upon the tubular secretion. In the next video, we will see some other points about the human excretory system. Thank you.